This is Professor Will, CBMD, PhD. Hey, Sunday, May 27, 2018, is 14:58 and 9 seconds Eastern Standard Time. Science News. In a break with dogma, myelin boosts neuron growth and spinal cord injuries. The molecules inhibits adult axon regeneration, but appears to stimulate young neurons. Date, May 23, 2018. Source, University of California, San Diego. Summary: researchers report that adult rat myelin actually stimulated axonal outgrowth in rat neural precursor cells, NPCs, and human-induced pluripotent iPSC-derived neurostem cell, NSCs. But in a new paper published in the May 23rd online issue of Science Translational Medicine, researchers at the University of California San Diego School of Medicine report that adult rat myelin actually stimulated axonal outgrowth in rat neural precursor cells, NPCs, and human-induced pluripotent iPSC-derived neurostem cells, NSCs. It is really a remarkable finding because myelin is known to be a potent inhibitor of adult axon regeneration, but that isn't the case with precursor neurons or those derived from stem cells. Colleagues in Germany and Singapore monitored neurite outgrowth from NPCs and NSCs growing on a myelin substrate in Petri dishes. Neurites are projections from the cell bodies of neurons, either axons which carry signals outward to other neurons or dendrites which receive the signals. In both cases, they found outgrowth enhanced threefold. In subsequent studies using rats with spinal cord injuries, the researchers found that rat NPCs and human iPSC-derived NSCs implanted at the injury site both extended greater numbers of axons through adult central nervous system white matter and through gray matter, and preferentially associated with rat host myelin. Paring away some of the myelin molecules known to strongly inhibit axonal growth, colleagues identified a molecule called Neuronal growth, Regulator 1, and NEG1 as potential mediator between axons and myelin, permitting the former's growth. NEG1 is involved in the process by which cells attach a neighboring cell and interact. The growth factor plays an important role during embryological development when neurons are growing rapidly, but before myelin begins to have an inhibitory effect. When we implant neurostem cells into sites of spinal cord injury, they extend tens of thousands of axons out of the injury site for distances up to 50 millimeters. Adult axons, on the other hand, when coaxed to grow, extend 100 axons for a distance of up to 1 millimeter. These findings identify why axon outgrowth from neurostem cell implants is so much better than injured adult axon. The findings support the developing approach of using neural precursor cells and iPSC-derived stem cells as a viable and promising method for repairing spinal cord injuries. More specifically, they point to the need to further investigate the stimulatory effect of myelin on NPCs and NSC, which could potentially be exploited for neural repair after spinal cord injury. Good day, this is Professor Will, CBMD, PhD. Hey, Sunday, May 27, 2018 is 15:15, 28 seconds Eastern Standard Time. Science News. Study uncovers cause of pesticide exposure, Parkinson's link. Low-level exposure to pesticides disrupts cell in a way that mimics the effects of mutations known to cause Parkinson's disease. Date, May 23, 2018. Source, University of Gulf. Summary: previous studies have found that an association between two commonly used agrochemicals, paraquat and MANAB, and Parkinson's disease. Now a professor has determined that low-level exposure to the pesticides disrupts cells in a way that mimics the effects of mutations known to cause Parkinson's disease. Adding the effects of the chemicals to a predisposition to Parkinson's disease drastically increases the risk of disease onset. Previous studies have found an association between two commonly used agrochemicals, paraquat and MANAB, and Parkinson's disease. Researchers has determined that low-level exposure to pesticides disrupts cells in a way that mimics the effects of mutations known to cause Parkinson's disease. Adding the effects of chemicals to predisposition for Parkinson's disease drastically increases the risk of disease onset. And people exposed to these chemicals are about 250% higher risk of developing Parkinson's disease than the rest of the population. We wanted to investigate what is happening in this susceptible population that results in some people developing the disease. Used on a variety of Canadian crops, paraquat is used on crops as they grow, and MANAB prevents post-harvest spoiling. Published in the Journal Federation of American Societies for Experimental Biology, this study used stem cells from people with Parkinson's disease that had a mutation in a gene called synuclein that is highly associated with increased risk of Parkinson's disease, as well as normal embryonic stem cells, in which the risk-associated mutation has introduced by gene editing. Until now, the link between pesticides and Parkinson's disease was based primarily on animal studies as well as epidemiological research that demonstrated an increased risk among farmers and other exposed to agricultural chemicals. We are one of the first to investigate what is happening inside human cells. From the two types of stem cells, research teams made dopamine-producing neurons, the specific neurons affected in Parkinson's disease, and exposed them to two agrochemicals. In exposing cells to agrochemicals, energy-producing mitochondria were prevented from moving to where they were needed inside the cell, depleting the neurons of energy. Neurons from the Parkinson's patients and those in which the genetic risk factor was introduced were impaired at doses below the EPA reported lowest observed effect level. Higher doses are needed to impair function in normal neurons. People with predisposition for Parkinson's disease are more affected by these low-level exposure to agrochemicals and therefore more likely to develop the disease. This is one of the reasons why some people living near agricultural areas are at higher risk. Research findings indicate that we need to reassess current acceptable levels for these two agrochemicals. The study shows that everyone is not equal, and these safety standards need to be updated in order to protect those who are more susceptible and may not even know it. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, 15:30, 29 seconds, Eastern Standard Time. Science News. Space-like gravity weakens biochemical signals in muscle formation. Microgravity conditions affect DNA methylation of muscle cells, slowing their differentiation. Date, May 23, 2018. Hiroshima University. Summary. Microgravity conditions affect DNA methylation of muscle cells, slowing their differentiation. Astronauts go through many physiological changes during their time in spaceflight, including lower muscle mass and slower muscle development. Similar symptoms can occur in the muscles of people on Earth's surface, too. In fact, it could affect everyone to some extent later in life. Age-related skeletal muscle disorders such as sarcopenia are becoming a greater concern in society. It is especially a big concern in Japan, where the number of aging people is increasing. In a study published in Microgravity, a medical research group at HU shed light on these similarities. They found that the process that affects gene expression of differentiating muscle cells in space also affect cells in the presence of gravity. The genetic and molecular basis of impaired muscle development has been unclear. There is a pressing need to understand it and come up with better treatment outcomes. Research teams investigated how simulated microgravity, that is, gravity in space-like conditions, affects muscle cell differentiation and gene expression. They observed what happened to rat muscle cells over time. Some cells were treated with a drug that stops DNA methylation from happening, while other cells were not. DNA methylation is a process that controls gene expression and muscle cell differentiation. Next, they grew the cells either in normal gravity or inside Gravite, a machine that simulates gravity at levels that astronauts experience in spaceflight. Cells in microgravity exhibit less cell differentiation after all. However, cells growing without the drug formed muscle fibers at a slower rate and showed less gene expression. One gene, MyoD1, was of particular interest. Its expression levels were significantly lower in microgravity conditions than when growing with the drug that stopped DNA methylation. Within gravity as well as without it, the group concluded that DNA methylation appears to be a key player in regulating muscle cell differentiation. These findings highlight genes affected by DNA methylation, like MyoD1, as potential targets for treating patients with skeletal muscle atrophy. The team's results can be used to utilize experiments where muscle atrophy of astronauts uses myotubes because it is easy to understand morphologically. Additionally, the findings of this epigenetics can be used in many differentiated cells, stem cells, or cancer. The Micro G Center, the Kennedy Space Center of NASA, where Research Advisory Committee member and NASA have already conducted experiments to cultivate stem cells on the International Space Station, where this paper can also provide insight. Research teams are expected to start a massive space experiment at NASA Center for Advancement of Science in Space, CASIS. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, is 15:44, 33 seconds, Eastern Standard Time. Science News. Flow of cerebrospinal fluid regulates neural stem cell division. Date, May 18, 2018. Source, German Research Center for Environmental Health. Summary. Stem cells in the brain can divide and mature into neurons participating in various brain functions, including memory. In a new study, scientists have shown how this works. They found that ion channels play a key role in mediating force signals to the neural stem cells to activate them. The ancient Greek aphorism Pantaria means everything flows, a phrase used by philosophers to describe the constant flux and interplay between the past and renewal. Researchers discovered that the flow of cerebrospinal fluid is a key signal for neural stem cell renewal. Neural stem cells in the brain can divide and mature into neurons, and this process plays important roles in various regions of the brain, including olfactory, sense, and memory. These cells are located in what is known as the neurogenic stem cell niche, one of which is located at the walls of the lateral ventricles, where they are in contact with circulating cerebrospinal fluid. The cerebrospinal fluid fills the brain, and its roles are still ill understood. This highlights the role of the fluid as a key signal, but this time not a chemical, but a physical signal. Researchers discovered that brain stem cells are also influenced by physical forces of this fluid flow. Channel protein plays a key role. The whole mechanism is controlled by the ENAC molecule. The abbreviation stands for epithelial sodium Na channel and describes a channel protein on the cell surface through which sodium ions stream into the cell's interior. We were able to show in an experimental model that brain stem cells are no longer able to divide in the absence of ENAC. Conversely, a strong ENAC function promotes cell proliferation. Further tests showed that the function of ENAC is augmented by shear forces exerted on the cells by cerebrospinal fluid. The physical stimulation causes the channel protein to open for longer time and allow sodium ions to flow into the cell, thus stimulating division. The results came as a big surprise since ENAC had previously only been known for its functions in the kidneys and lungs. Research teams now want to better explore the fast signals mediated by ions in stem cells and clarify the extent to which the findings are also relevant to treatment. After all, pharmacological ENAC blockers are already used clinically to relieve certain types of hypertension. Now it is known that they can also influence stem cells in the brain and thus brain functions. Here too, research is in constant flow. Pantaria. Notes: the ventricular system is a set of 4 interconnected cavities, ventricles in the brain where the cerebrospinal fluid CSF is produced. This is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, at 16:21, 18 seconds, Eastern Standard Time. Science News. Immune cells provides cradle for mammary stem cells. Study shows how macrophages and mammary stem cells talk to each other through chemical signaling. Date, May 17, 2018. Source, Princeton University. Summary: researchers have made new discoveries about how an immune cell, known as the macrophage, which normally fights infection by swallowing foreign invaders, nurtures mammary gland stem cells through a chemical signaling molecule. The study may provide important clues about the roles of macrophages in breast cancer progression. The new study finds that one of the toughest characters in the immune system, the macrophage, has nurturing side, at least when it comes to guarding the developing breast. Learning more about the factors that keep mammary stem cells alive and healthy may provide insights into the development of breast cancer when such regulation goes awry. Stem cells are cells that have the potential to mature or are differentiated into several different types of cells. Mammary stem cells shepherd breast development during puberty and are active during pregnancy and lactation. The team found that the macrophage, a type of white blood cell whose name translates to big eater because it fights infection by engulfing foreign invaders, helps create a special environment known as the stem cell niche that keeps stem cells in their premature state and able to duplicate and differentiate into mature cells when needed. Stem cells come in various types, from ones that are already committed to develop into certain organs as the mammary glands to ones that can form nearly any cell in the body. These cells may also become cancerous, leading to tumors and eventually to the spread of cancer to other organs. To better understand how mammary stem cells regulate breast development and how they might turn cancerous, the researchers studied how macrophages interact with mammary stem cells via a molecular signaling mechanism. These signaling mechanisms involve a protein called Notch, which acts as a receptor for a signaling molecule called DLL1. Previous research in the lab found that mammary stem cells make a lot of DLL1. In the current study, researchers knocked out the DLL1 gene in mice and found that there is a significant delay in breast development during puberty as well as delayed breast expansion during pregnancy and lactation. Furthermore, the number of mammary stem cells was reduced both during mammary gland development and during pregnancy. Meanwhile, mice with the DLL1 gene intact contained mammary glands with a greater ability to regenerate and to differentiate into mature mammary cells compared to the DLL knockout mice. The mice lacking DLL1 also had fewer macrophage cells in their mammary glands. The researchers found that the blinding of DLL1 to Notch2 and Notch3 receptors is important for keeping the number of macrophages high in the mammary stem cell niche, and important for keeping the mammary cells in their stem cell state. The researchers also found that the presence of DLL1 is linked to the expression of genes for another signaling pathway involving a molecule called WNT. The expression of DLL1 has caused the macrophage to express genes for WNT3, 10, and 16, which further enhance the stem cell behavior of the mammary stem cells. When the researchers used genetic or chemical methods to inactivate the macrophages in the mammary glands of mice, the mammary stem cell activity decreased. We conclude that the signaling molecule DLL1, which is produced by a mammary stem cell, activates the signal of macrophages to sustain mammary stem cells. The survival and function of these mammary stem cells and macrophages are dependent on each other, and they communicate through these cellular messaging systems. The Notch WNT crosstalk that occurs at the mammary stem cell niche is crucial for the development of mammary gland. Research crosstalk may have implications in breast cancer, as both these signaling pathways are aberrantly increased in breast cancer. Researchers expand our knowledge about the role of macrophages in mammary gland development, first demonstrated by other research groups almost 2 decades ago, and more recently by researchers who found that macrophages influence stem cell activity in the mammary gland. The study also raises several questions about the specific roles of multiple WNT molecules expressed in mammary cells and surrounding cells, as well as in tissue-resident macrophages in mammary stem cells. These studies on normal mammary gland development may provide important clues about the roles of macrophages in breast cancer progression. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, 16:53:23 seconds Eastern Standard Time. Science News. Signal surface protein boosts multiple oncogenic pathways in acute myeloid leukemia. Date, May 17, 2018. Source, Rockefeller University Press. Summary: researchers have discovered that a signaling protein elevated in patients with acute myeloid leukemia AML plays a much wider role in the disease than previously thought. The study raises hopes that current efforts to target this signaling protein could be a successful strategy to treat AML and other blood cancers. AML is driven by leukemia stem cells that differentiate abnormally and proliferate excessively, eventually displacing normal healthy blood stem cells from the bone marrow. Leukemia stem cells are usually resistant to traditional chemotherapies, and so researchers are trying to develop new, more targeted approaches to eliminate these crucial cells. One possible target is the protein called interleukin-1 receptor accessory protein, IL-1RAP, which is often highly expressed on the surface of the leukemia stem cells but largely absent from normal blood stem cells. But whether leukemia stem cells require IL-1RAP to survive and proliferate, and therefore whether inhibiting IL-1RAP could be a successful way to treat AML, is unclear. A team of researchers led by the Albert Einstein College of Medicine found that antibodies, small hairpin RNAs, or genetic deletion targeting IL-1RAP induced the death of AML cells, including leukemia stem cells isolated from patients. The effects were seen in the absence of immune effector cells, indicating that AML cells intrinsically depend on IL-1RAP. In contrast, antibodies targeting IL-1RAP had no effect on the growth and survival of normal blood cells. Antibody treatment also inhibited the proliferation of AML cells in mice without causing any negative side effects. As its name suggests, IL-1RAP works in conjunction with the interleukin-1 receptor to promote an inflammatory signaling pathway that could play a key role in the development of AML, but colleagues found that IL-1RAP also enhances the activity of two other membrane receptor proteins, FLT3 and CKIT, that are known to stimulate the proliferation of leukemia stem cells when activated by their ligands. IL-1RAP antibodies inhibited the ability to these ligands to induce proliferation of AML cells. Our findings show that IL-1RAP can amplify multiple key pathways in AML, demonstrating a much broader role for this protein in disease pathogenesis than previously appreciated. Researchers are currently developing ways to prompt a patient's own immune cells to selectively recognize and kill IL-1RAP expressing cancer cells, but AML patients generally have effective immune systems. This new research suggests that directly targeting IL-1RAP with antibodies or specific drug molecules could be even more effective at killing AML cells. The findings also suggest that IL-1RAP-directed immunotherapies could be even more promising than currently assumed, because AML cells functionally depend on the IL-1 RAP, they cannot simply down-regulate the protein to escape immunotherapy and are therefore less likely to develop therapeutical resistance. Importantly, as IL-1 RAP is also overexpressed in the stem cell of chronic myeloid leukemia and high-risk myeloid dysplastic syndromes, there is significant therapeutical potential in further developing IL-1 RAP-directed targeting strategies. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, 17:34, 40 seconds Eastern Standard Time. Science News. Researchers take key step toward growing human organs in laboratory. Date, May 16, 2018. Source, Oregon State University. Some researchers have learned that precursor cells for skeletal muscles actually also give rise to neurons, blood vessels, blood cells, and immune cells, pushing science one step closer to generating body parts in the laboratory. Combining developmental biology, genetics, and bioinformatics, scientists at Oregon State University confirmed that PAX3 plus cells act as a multifaceted stem cell niche for multiple organs at embryonic stages. We now have the ability to label cells based on expression profiling of sequence-specific transcription factors. We can give a molecular code to each cell to make it distinct in time and space. During the progression of a cell, the code changes. That's beautiful because now we can discover niches, stem cell pockets, and we can use this information to fix so many genetic or environmentally caused disorders. This gives us the possibility to make an entire cell lineage or an entire organ in a petri dish. In a mouse embryo model, state colleagues of veterinary medicine and engineering compared gene expression profiles of the PA*3 plus cell population over several days. They isolated lineage trace cells from 4 limbs at different embryonic days and performed whole transcriptome profiling via RNA sequencing. That let us identify genes involved in the skeletal, muscular, vascular, nervous, and immune systems, of all which go into making a functional limb. Expression of genes related to the immune, skeletal, and vascular systems showed big increase over time, which suggests PA*3 plus cells give rise to more than muscles. They are involved in patterning in the three-dimensional formation of the forelimb through multiple systems. Research opens the potential to use stem cell pockets, for example, to grow a new arm or leg or other organ, for example, who's lost a body part to accident or disease. That cell population can give rise to so many different cell types. We can use it at the development stage and allow it to become something else over time. It is a great source we have to fix different organs with issues from injury, toxicity from drugs, anything that can generate cell death in any part of our body. A genetic program that runs during embryogenesis normally generates adult cell types. We can identify these cells and be able to generate not only 1 but 4 different organs from them. This is a prelude to make body parts in a lab. Researchers liken the research to a mechanic taking apart an engine down to every last nut, bolt, and washer. An engine has so many little parts, and you can't fix a broken engine if you don't know what all these little parts do. Likewise, you can't make a limb if you don't know about all the cell parts within the limb. You can't use only bone or only muscle or only veins. You need everything working together. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, 17:54, 16 seconds Eastern Standard Time. Elastic microspheres expand understanding of embryonic development in cancer cells. Date, May 14, 2018. Source, University of Illinois at Urbana Campaign. Summary. A new technique that uses tiny elastic balls filled with fluorescent nanoparticles aims to expand the understanding of the mechanical forces that exist between cells. Researchers report. The team has demonstrated the quantification of 3D forces within cells living in petri dishes as well as live specimens. This research may unlock some of the mysteries related to embryonic development in cancer stem cells, i.e., important example, tumor repopulating cells. For decades, scientists have struggled to quantify the forces called tractions that push, pull, and squeeze cells throughout their life cycles. The tools available to measure force were not small enough to fit into intracellular spaces or sensitive enough to detect the many-scale movements within cell colonies. Although small on a human scale, the magnitudes of these mechanical forces are far from trivial at the cellular level. According to the new study, prior research by the Illinois group and others indicates that traction plays a fundamental role in cell physiology. If we place a single cell in a medium within a petri dish, it will not survive for long, even if we provide all of the nutrients needed. The cells fail to form any sort of tissue because there is no support for scaffolding on which to build. As cells grow and reproduce, they exert forces on each other while competing for space. The team found that if they inject their tiny elastic spears into early-stage embryos of zebrafish and colonies and melanoma cells of mice in petri dishes, they too experience the forces. The cells do not seem to mind the intrusion. The spears are made of non-toxic microgel, and even though the cells will push them around, they do not seem to interfere with development. To measure the amount of force imposed on the cells, the team placed fluorescent nanoparticles inside the spears. When the cells squeeze the spears, the nanoparticles all move the same amount per area force. The researchers can then measure the motions of the glowing particles using fluorescent light microscopy to calculate the amount of force extended on the spears and cells. Using this technique, the team has marked the first successful measurement of all three types of force: compression, tension, and shear, in all three dimensions. This ability to quantify force in cells may be very important to cancer cell research. The team found that when melanoma tumor cells of mice in vitro began to reproduce from a single cell to about 100 to 200 cells, compressive stress does not increase. We thought that cancer cells would generate more pressure at this early growth stage while the mass of the tumor increases, as we observed in zebrafish embryos, but they do not. We suspect that the cancer cells begin to spread out or metastasize right after this stage. Primary tumors are usually not deadly. The real killer appears to be the spread of tumor repopulating cells from primary tumors into soft tissues with low intercellular attractions, like bone marrow, brain, lung, and liver. Although the underlying mechanism for metastasis is unclear, we have hypothesized that tumor repopulating cells spread very rapidly in these secondary soft tissues. Having the ability to measure changes in tractions at the intercellular level may serve as an early cancer detection tool. The microgel spear technology may also help unravel the mechanisms behind the metastasis-halting synthetic drug recently described by research and colleagues. In addition, authors are continuing to apply this technology to stem and embryonic cell research. When other researchers see this powerful new tool that we have developed, they will be excited to use it in many different cell physiology development and disease applications. Good day, this is Professor Will, CBMD, PhD. Hey, Sunday, May 27, 2018, 18:11, 37 seconds, Eastern Standard Time. Science News. Molecule that supports blood cell production under dietary stress is identified. Date, May 7, 2018. Source, Kanazawa University. Summary: researchers report how the SPRED1 molecule is involved in hematopoietic stem cell self-renewal. Experiments with mouse models show that under normal conditions, SPRED1 acts as a negative regulator, while under diet-induced stress, it protects hematopoietic homeostasis. The researchers looked at SPRED1 protein because it binds to CKIT, a molecule involved in signaling processes that govern HSC development and regulation. Experiments with SPRED1 deficient mice showed that the protein is not crucial for normal hematopoiesis, the formation of the cellular components of blood, in stress-free situations. Furthermore, SPRED1 deficiency promoted HSC self-renewal, resulting in prolonged cellular lifespan, increased competitiveness, and better resistance of physiological stress. A pathological condition caused by mutations in the SPRED1 gene, they found that SPRED1 deficiency does not lead to the development of leukemia, and concluded that the protein is therefore not a conventional tumor suppressor. In wild-type mice aging transplantation and treatment with lipopolysaccharide, a procedure mimicking bacteria infection, unregulated SPRED1 protein, the scientists suggest that this unregulation may lead to HSC dysfunction in conditions of physiological stress. However, when the researchers looked at the effect of high-fat diet on SPRED1 deficient mice, they found that the diet triggered the development of a particular type of blood cancer. This finding clearly demonstrates that SPRED1 does play an essential role in regulating hematopoietic homeostasis. The study highlights the complex relation between SPRED1 and hematopoiesis, establishes a link between the function of the protein and dietary stress. Regarding future research, and scientists conclude that investigation of the pathophysiological role of dietary factors in stem cell self-renewal and exploration of approaches that manipulate SPRED1-mediated control of HSC self-renewal may uncover innovative technologies for preventing diet-related diseases and malignancies. Hematopoiesis is spread 1. Hematopoiesis is the process of the formation of cellular components of blood. All blood cellular components are generated from hematopoiesis stem cells, HSCs, and HSC reside in bone marrow, and can differentiate into all types of blood cells, red blood cells, lymphocytes, and myeloid cells. When they differentiate, some of the produced cells are again HSCs, so there is always a supply of HSCs, the self-renewal process. Kanazawa University have studied the relationship between a protein called spread1, sproutly related EVH1 domain containing protein 1, and the self-renewal of HSCs in mouse models. On the other hand, they found that under normal conditions, spread1 acts as a negative regulator for HSC self-renewal, while on the other hand, in a situation of diet-induced stress, it safeguards hematopoietic hemostasis. Legius syndrome is a condition resulting from mutations of the spread1 gene. Symptoms include freckles, learning disabilities, and attention deficit hyperactivity disorder, ADHD. It is often confused with neurofibromatosis type 1, NF1, and later tumor growths are common, whereas in Legius syndrome they are absent. The connection between spread1 deficiency and Legius syndrome, and found that spread1 does not play the role of the conventional tumor suppressor. Today's is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, 18:26, 43 seconds Eastern Standard Time. Science News. New parts of the brain become active after students learn physics. Date, May 24, 2018. Source, Drexel University. Summary, a new study showed that when confronted with physics problems, new parts of a student's brain are utilized after receiving instruction in the topic. Parts of the brain not traditionally associated with learning science become active when people are confronted with solving physics problems, a new study showed. Using fMRI, functional magnetic resonance imaging, to measure blood flow in the brain, the researchers looked to map what areas become active when completing a physics reasoning task, both before a course on the concepts and after. The neurobiological processes that underpin learning are complex and not always directly connected to what we think it means to learn. More than 50 volunteer students took part in the study, in which they were taught a physics course that utilized modeling instruction, a style of teaching which encourages students to be active participants in their learning. Before they participated in the class, the students answered questions from an abridged version of the force concept inventory while undergoing fMRI. The force concept inventory is a test that assesses knowledge of physics concepts commonly taught in early college physics classes. After the volunteer students completed their physics course, they again took the force concept inventory, once more monitored by fMRI. In the pre-instruction scans, part of the brain associated with attention, working memory, and problem solving, the lateral prefrontal cortex and parietal cortex, sometimes called the brain's central executive network, showed activity. One of the keys seemed to be an area of the brain, the dorsal lateral prefrontal cortex, that generates mental simulations. This suggests that learning physics is an imaginative process, which is not typically how people think of it. After the subjects had completed their class, comparison of the pre- and post-learning scans revealed increased activity in the frontal poles, which was to be expected since they've been linked to learning. But there was another area that also became active, the posterior cingulate cortex, which is linked to episodic memory and self-referential thought. These changes in the brain activity may be related to more complex behavioral changes in how students reason through physics questions post-relative to pre-instruction. These might include shifts in strategy or increased access to physics knowledge and problem-solving resources. One of the aims of the study was to further explore how the form of teaching used. Modeling instruction encourages students to use their own mental models to understand new concepts. The idea of mental models is something that people who research learning love to talk about, but have no evidence of what is happening inside brains other than what people say or do. We are actually looking for evidence from inside the brain. Researchers think their study provides a good look at what might be typical when these mental models take hold. But why physics? What makes this idea subject to study mental modeling in the brain? Researchers say there has been some research on the brain networks associated with learning math and reading, but mental modeling especially lends itself to physics, which has not gotten as much attention. Physics is a really good place to understand learning for two reasons. First, it deals with things that people have direct experience with, making formal classroom learning and informal understanding both relevant and sometimes aligned and sometimes contrasted. Second, physics is based on laws, so there are absolutes that govern the way the body works. Researchers Want to follow up on the questions of mental assimilations in physics to see where that shows up at different levels of physics, learning and with different populations. But this whole study opens up many new areas of investigations, and researchers are pretty excited about how it will play out. Today's is Professor Will, CBMD, PhD, Hayes, Sunday, May 27, 2018, 18:41, 33 seconds Eastern Standard Time, Science News. Reading the minds of pilots on the fly. Research paves the way to improve the safety and efficiency of aircraft-pilot interactions. Date, May 21, 2018. Source, Drexel University. Summary: wearable brain monitoring sensors allowed researchers to measure cognitive workload while aircraft pilots completed memory tasks. After a plane engine blew apart at 32,000 feet in the air last month, the pilot flying Southwest Flight 1380 safety brought the Boeing 737 to an emergency landing in Philadelphia. Consider what was racing through her mind: where to land, the speed and altitude to maintain, how to aid an injured passenger, and comfort a bewildered crew. It is likely Schultz was experiencing cognitive overload. The efficiency and safety of human-machine systems depend on cognitive workload and situational awareness of human operators. Unfortunately, many human-machine interfaces expose users to workload extremes, diminishing the operator's attention and potentially leading to catastrophic consequences. An ideal human-machine system would actually be able to read its operator's mind in real time, to know how well he or she was paying attention or able to process new information. Such a system may sound like the makings of a sci-fi movie. Researchers successfully measured the brain activity of pilots in real time using functional near-infrared spectroscopy, or FNIRS. The results were published in this week, Frontiers in Human Neuroscience. Unlike traditional functional magnetic resonance imaging, fMRI, in which a person is resigned to a large machine while lying down, Drexel's portable FNIRS system is worn like a headband, making it ideal for measuring brain activity while studying subjects move around freely in their natural environments. The FNIRS system measures the brain at work by monitoring blood oxygenation changes in the prefrontal cortex, the area underneath the forehead that is involved in cognitive functions such as problem-solving, memory, judgment, and impulse control. When first learning a new task, for instance, this area of the brain is highly activated. However, as you become more proficient, these tasks move to other brain areas, clearing up important resources in the prefrontal cortex for skills like split-second decision-making. The exciting thing is we can now quantify this in a flying scenario. Research envisions that the airplane itself might one day be able to assess the cognitive and emotional state of the pilot, and then make adjustments accordingly. In the future, understanding the underlying neurocognitive process of pilot-plane interactions could help to make simulators more realistic, as well as to improve the safety and efficiency of aircraft-pilot interactions. We believe that this type of approach will open a whole new direction of research for studying parameters in an aviation setting and eventually designing better machines.